Okay guys, I am here to review the most missed questions for the PPCs on Unit 3 to help you study for your test. This set of PPCs was short and it seemed to focus more on enzymes, so make sure that you're looking at your photosynthesis and respiration practice questions too. The first question that the majority of us missed was this question on the peroxidase enzyme, and it's not that you guys don't understand enzymes. This is actually a question about negative control, and I think that's where the misunderstanding is. Okay, it says researchers are investigating the influence of environmental pH on the activity of peroxidase, an enzyme that catalyzes the conversion of hydrogen peroxide to water and oxygen gas. That should sound familiar. This is like the experiment we did in class, but in animals, the enzyme is catalase, and in plants, it's peroxidase. So it's very common to see this question where it's oxygen being measured. It's a common enzyme experiment. So it says the researcher included guaiacol, which is again what we use too, because it caused the solution to change color as the reactions proceeded, which the researcher relied on for measuring the reaction rate. So indirectly, he measured the reaction rate with absorption, okay? And then finally, the researcher added the same amount of peroxidase to each test tube and measured the rate of the reactions at 23 degrees. And these are our results, okay? But the part that we were unsure of is the negative control. If you're asked for a negative control, that means this is a control where you do not expect a result. You expect no activity. Okay, so whenever you see negative control, that means a control where there's going to be no reaction. So if you look at your answers, 2 of you picked repeating the experiment at 25 degrees. That would not give you a negative control. You would still have a reaction. That's just changing an environmental factor. It's not adding a control. Repeating the experiment using twice the amount of hydrogen peroxide. Again, that would not be adding a control. It's just changing another factor, changing an environmental factor. And then repeating the experiment using twice the amount of peroxidase, same thing, that's changing the enzyme. So your answer is D, repeating the experiment using a heat denatured peroxidase. Okay, why is that your negative control? Because if it's denatured, it's an inactive enzyme, it's not going to work. So again, negative control means a control where you expect no result. Okay, question 3 was also about this same experiment, but again, it's not really an enzyme question. It's more of a question about experimental design. Okay, so the researcher is proposing to use oxygen gas to measure the reaction rate, and it wants to know which justifies the use of this modification for creating an appropriate control. And so you've got to remember that a control in an experiment is something that allows you to eliminate variation or eliminate error in your experiment. So that's what we're looking for. Okay, we're looking for something that can eliminate error or differences, variation in our experiment. So number 1, it can be repeated without hydrogen peroxide, which will help eliminate an uncontrolled variable. If you have no hydrogen peroxide, you're just not going to have a reaction. Just because it says it will eliminate an uncontrolled variable doesn't mean that it does. There's really no purpose in running the experiment without hydrogen peroxide because you just would not get oxygen at all. The source of your oxygen is the hydrogen peroxide. So using oxygen gas is not compatible with this. Okay, the experiment can be repeated without peroxidase, which will introduce a second dependent variable. First of all, eliminating a second independent variable is not the same as adding an appropriate control. So that's the first part of that that's wrong. And again, if we repeat the experiment without peroxidase, that doesn't explain how using oxygen gas is going to help us measure reaction rates. Because without the enzyme, I just won't have any oxygen. Okay, your answer is C. The experiment can be repeated without guaiacol, which will reveal the effect of guaiacol on the reaction rates. Remember that guaiacol is a oxygen indicator, but it could also have potentially side effects that we don't know about or variations that we don't know about in our data. So it's good to run an experiment where we can measure oxygen directly to eliminate the effect of guaiacol. Remember guaiacol. When we are measuring with guaiacol, it's an indirect measurement of oxygen. And again, repeating the experiment without water, it doesn't really explain how using oxygen gas relates to that. So that's question 3. Okay, question 4 is about acetylcholine concentration, and it tells you up here, acetylcholine is a protein that catalyzes the conversion of acetylcholine to acetate in choline. When the concentration of ACH in an aqueous solution is held constant, the rate of the reaction catalyzed by acetylcholine increases with increasing concentration. At low concentrations, a small increase in the substrate concentration results in a large increase in reaction rate. Okay, and then at high concentration, a large increase in substrate only results in a small increase in the reaction rate. So what's your correct observation? A lot of these you can just eliminate because they don't match up with what the experiment is saying. So ACHE, that's your enzyme, begins converting product into substrate as the acetylcholine concentration changes from low to high. Enzymes aren't regulated by the concentration of their substrate. Enzymes are going to work on all substrates present. They can be regulated by allosteric molecules, but not by their own substrate. So that's why it's not B. It becomes denatured as the acetylcholine concentration changes from high to low. Remember, acetylcholine is your substrate, and a substrate is not going to denature your enzyme. The only two things that can denature your enzyme are heat and pH. And the activity levels can be changed by changes in temperature, pH, or the presence of an inhibitor or a stimulator. Your answer is A. The active site is specific for acetylcholine. And only, this is the part that makes it true, only one substrate molecule can occupy the active site at a time. So when there's already an acetylcholine molecule in an enzyme, it can't move any faster. So at high substrate concentration, all of your enzymes are working. There are no unoccupied enzymes. So adding additional substrate won't help. That's the graph you guys have seen that looks like this. Okay, you're increasing and then it levels off. So this would be like activity here on the Y, and this would be substrate concentration. When your substrate concentration reaches a certain point, all enzymes are occupied. It can't work any faster. The only thing you can do to overcome that is adding additional enzyme. And then Q14, a lot of us miss Q14, but it's not going to be on the test. We're not talking about the law of thermodynamics right now. Okay, Q15, brown fat. We did not get to talk about this, but there are many mammals who have brown fat that help maintain temperature. And basically what happens is the fat is burned and heat is released instead of ATP. I think a lot of us looked at this and we were like, I don't know what brown fat is. So we just kind of guessed. But everything that you need is really in your diagram. Remember, if you have a diagram, don't panic. Try to find what you know. You've got your ATP synthase being ran by, again, a hydrogen gradient. This is oxidative phosphorylation. Okay, so this part looks the same as other respiration, but right here is where things get different. You have an uncoupling protein, and what's coming out of that is heat. So this diagram is showing you that at the end of the process, the excess energy is being released as heat. And you can see that your hydrogen ions are moving out. Pretty much everything you need is on this particular protein. The electron transport chain is running. I'm synthesizing ADP to ATP, but I have this special protein that's releasing hydrogen out of the cell, and in the process, it releases heat. And that's given to you up here. The uncoupling protein allows hydrogen ions to leak from the inner membrane space back into the matrix. Remember, losing that proton gradient, you have to know what that does. If you lose a proton gradient, it's going to affect the amount of ATP that you're able to make. However, okay, you still have energy from those electrons that has to go somewhere. And in this case, you can see right here that it's being released as heat. But you still want to read and eliminate your answer choices. The uncoupling protein increases the production of ATP and causes more body heat. Well, ATP is produced over here, and the heat that's being released is all the way over here, separate. My uncoupling protein is not involved with ATP synthase in my diagram. B, the uncoupling protein reduces the proton gradient and produces heat to warm the animal without ATP production. Okay, so the way you find that, this is releasing hydrogen. Okay, that's relieving my gradient. It's taking away my gradient. You guys know that if I take away the hydrogen gradient, I can't have ATP synthase running because it's powered by that. And then you see heat coming out on the other side of your uncoupling protein. So that's how you know. That's how you know that it's your heat that's being released here. All right, so your answer is B. But 4 of you did pick C, so I want to explain why it's wrong. The uncoupling protein causes an increase in the proton gradient, which causes more ATP to be produced that helps to warm the animal. Oops, sorry. Okay, so here's the issue. Like, you maybe did think ATP can help keep animals warm because we haven't talked about it. But the problem is, it says that the uncoupling protein is going to increase the proton gradient. And if you look up at your diagram, it's releasing hydrogen out of the cell. It's moving the opposite direction of the proton gradient. So even if you're not sure about brown fat, you can eliminate from looking at your diagram. It's not C because the uncoupling protein is reducing my gradient. And then D, the uncoupling protein in the tissue reduces the creation of ATP. And again, here's your problem. Creates an increase in the proton gradient. Okay, that is the part of it that even if you have no idea what's going on with that question, you can look and see that that uncoupling protein is actually decreasing the proton gradient. This question 17, it's an evolution question. It is included in this unit, but I kind of like to talk about all this stuff at the end. So this will not be on your test, and we'll hit this question again when we get to ecology and evolution. What best clarifies how the data support the research? And we first, like, if we're going to justify a claim or support a claim, we need to understand what the claim is. So a researcher claims that a certain herbicide suppresses plant growth by inhibiting chloroplast function. So if I'm inhibiting chloroplast function, okay, that's going to be photosynthesis. I don't know which part yet, but that's what my table is going to help me with. Okay, so here's my herbicide concentration. And notice, this is really important, and we haven't talked enough about this. My pH difference is dropping. Okay, pH is a measure of hydrogen concentration. Okay, so if my difference in pH is dropping, then what that means is my difference in hydrogen concentration is also dropping. So let me say that one more time. If you don't recall from chemistry, pH is a measure of hydrogen concentration, hydrogen ion concentration. So if the difference, this isn't a raw measure of pH, it's a measure of the difference in pH across the membrane. So as the difference falls, what that means is my concentration gradient is getting smaller. And remember, without a gradient, I can't make ATP. So that's why your ATP goes from very high when I have a big gradient to very low when I have no gradient. So which clarifies, let's see what you guys picked. A lot of you guys picked B, but on A, the thylakoid membrane is more permeable to carbon dioxide than to polar molecules. Here's the problem, and this is what I really want you guys to see, is even if you know nothing about the question, or even if you're not sure whether the answer is right, sometimes you can eliminate, but by just looking at what two things have been put together. Okay, so this answer choice puts together the thylakoid membrane and carbon dioxide. Carbon dioxide is part of the Calvin cycle. So I can eliminate that question just because these two don't go together. Okay, again, the majority picked the right answer. ATP synthase activation depends on a proton gradient. That's what's showing us here. A difference in pH means a difference in proton gradient. And then one person picked D, carbon fixation takes place in the stroma. That doesn't really make sense because my question is about the thylakoid. So big take home point from this question is that difference in pH is the same thing as hydrogen concentration. Question 22 is another one that a lot of us missed, but it's ecology and evolution again. So we will talk about that in another unit. Remember to review your respiration questions, and I will send a key out for that later this weekend.